Let's now walk through another key feature of the cell annotation platform, which is the ability for researchers to do manual cell annotation directly via their web browsers. So as opposed to working with R or Python to iteratively analyze the data, which is normally, as I say, it's an iterative process. You have to select cells, calculate gene expression, do this again and again, and create multiple plots, which creates reproducibility issues. We've tried to create this easy-to-use web browser such that scientists can work directly via the molecular data and create cell annotations. And they would do this for two reasons primarily. One is to share with their collaborators for review before publication with the public. And the second is to share these cell annotations with the scientific community for reviews. So in this case, we have a data set with over 400,000 blood cells. This is within a context of a study from 2020, 2021, looking at blood samples from SARS-CoV-2 patients. And they isolated the PBMCs, the peripheral blood mononuclear cells, from these blood samples. These are immune-specific cells, and integrated these together from these multiple patients to create this single data set. And in this case, we can see there's multiple sets of cell annotations already created by the team. These are very detailed cell annotations of the various types of T cells, various types of B cells, to very broad cell annotations. These are the very broad cell annotations. And we can click on these and look at the molecular signatures. But in this case, I want to walk through the case whereby we'll be creating a set of cell annotations from scratch. So in this case, I have edit access to the data set. I'm logged in, and I will create a new set of cell annotations. The description will be I'll create the set of cell annotations, and then the way this normally works is I'll select a set of cells, and the web browser is telling me that I have close to 70,000 cells selected, and in this case we'll run differential expression. Let me just start the process. I'm doing this against the background, and I'll explain what I'm doing. This is a standard procedure whereby the selected cells in red, I want to see which genes are more expressed or less expressed than some baseline. And the baseline is a population of other cells. So the standard thing to do within the field is to use other cells within the data set. And in this case, if there's over 400,000 cells, the assumption is that that should be enough statistical signal to measure what is special about these cells in red versus the background. And sure enough, we can see the results here that are loaded. We could look at a heat map to show what is special about these selected cells in red versus the others. I could zoom in if one needed to see really those specific cells, what are the gene expression patterns. But we could go back, and I happen to know some of these genes, and so PBMCs are often used within the field for tutorials within single cell analysis, and we have a lot of sort of priors regarding what some of these cell types could be. So for instance, we have LYZ here, which is a gene which encodes lysozyme. Lysozyme is used to break down cellular barriers. There's also CST3 here, which makes cystatin C, associated with kidney function. I happen to know that these are markers for dendritic cells, and so fair enough, I will create a cell label on these cells for dendritic cells. And you'll see there's a toggle here to do this. I can create a cell label, or I can mark these as doublets, which is a standard sort of sequencing error whereby two or more cells are sequenced within the same droplet. So this is a standard term within the field. Junk, these are cells which don't pass a QC threshold, or unknown, which is we just don't know. So I'll label these as dendritic cells. I know there's an existing ontology term for this. That's right, dendritic cells. I don't really like these synonyms, so I'm going to remove them. And I would say the closest category would be something like phagocytes. So phagocytes, there we go. Marker gene evidence, we just walked through this. It would be CST3 and lysozyme. And I would say that these are also in the canonical ways that I at least annotate these cells. I'm not sure if others do something different. There we go. And the rationale is looking at results against the background. Okay, and that will create a cell annotation. And so I'll be able to see that 16% of our cells were annotated. If I deselect those cells, I should be able to actually see that these are now colored in a certain way, and there's a label associated with them. And I could do this with other cells. We'll do one more, and I'll probably end the tutorial, but let's say we wanted to look at, let's say, these cells as well. So I'm going to create the selection icon. This is here. And so if I select, oops, my mouse is a bit tricky right now. Okay. Select these cells. These are 55,000 cells. We can do a different type of differential expression. As opposed to the background, I'll look specifically at these dendritic cells to show, to see what, oops. And we will do differential expression against the dendritic cells. That is the reference. And so, that was actually really quick. And yeah, sure enough, these are various genes which are coming up, which are known in the field for markers for a specific cell type, especially MS4A1, and I would say that, yeah, CD74, I think, and also CD37, CD70A, and you'll actually see this in the gene summary. The B lymphocyte antigen receptor, this is a surface marker for B cells. And so I will select these cells, and I will annotate them as B cells. So B cells, this is an ontology term. I know that B cells exist. These are some very broad cell types. Synonymous B lymphocyte in various ways, fair enough. I would say that lymphocytes would be a good category. Blood lymphocyte, and we go with lymphocyte here. Marker genes, it's basically everything we saw. So the big one would be MS4A1. There we go. Let's do CD79. What else did we see? We saw CD37. And for the big canonical marker, I would put down MS4A1 as a very broad marker for B cells. Rationale. We found genes MS4A1, CD78, and we can cap UI against dendritic cells. And that would create a cell label. Let me see, this is updated. And you can basically go through this entire set of cell annotations to provide these cell, you could go through this entire data set, excuse me, to provide these cell annotations. It's not existing within this data set, but often users provide other information that one could use to calculate these cell annotations. There's often a clustering that's saved within the data set, which didn't take place here, as it's an integrated data set. But based upon clustering of the cell annotations, you should be able to, as a user, select those various clusters and provide cell annotations. But in this case, which is with PBMCs, these are fairly classical markers, and so this is working out fairly well. Oh, there we go. Okay, so that is a walkthrough of how to start creating cell annotations de novo on your molecular data via the web browser. Thank you.